In this video, we'll be going into more depth about factors inside of cells that can make enzymes work faster or slower. Our target goals are marked 8.3 in this list. Pause the video and check them out. The factors that we're focusing on in this video are listed here. There are environmental factors like enzyme concentration, substrate concentration, temperature, and pH. We'll touch on cofactors and coenzymes a little bit, and then we'll spend a little bit of time on different types of enzyme inhibitors. While we explore how environmental factors affect enzymes, we'll be looking at graphs like this one. For each factor, I want you to recognize what's on the graph axes and the shape of the curve. The y-axis is going to have the reaction rate in each case, and the x-axis is going to show the different factor. So this one's going to change. Alright, first let's look at how enzyme concentration affects enzyme function. No matter what the factor we're looking at, there's two levels of understanding that I want you to be comfortable with. First, it's important to describe what happens to the reaction rate with these different conditions. In other words, look at the graph and tell me what's going on. Second, you have to explain what's happening at the molecular level to cause the differences that you see in reaction rate on the graph. With those two things in mind, let's check out what's happening here. So we've got enzyme concentration versus reaction rate, and you can see a couple of things going on. On the left side of the curve, over here, you can see that increasing the enzyme concentration causes an increase in reaction rate. But the right side of the curve tells a different story. At a certain level of enzyme concentration over here, adding more enzyme doesn't make the reaction rate go up. Why is that? Then that's the explain part. For the explain part, to explain why this is happening, I like to split the graph into two halves to describe what's going on with low enzyme concentration and high enzyme concentration. With low enzyme concentration, we can describe what's going on as when you increase the enzyme concentration, you're increasing the reaction rate. And the explain why this is happening is just simply that there's more successful collisions between the substrate and the enzyme. In order to get an enzyme catalyzed reaction, the substrate needs to actually touch with the enzyme. If there's more enzymes around, the chances that the enzyme is going to collide with the substrate will increase. But at some point, when you describe what happens with high enzyme concentration, the reaction rate plateaus or levels off. And that's just because the substrate becomes limiting. There's not enough substrate to keep the enzymes, I put in quotes, busy. In other words, what that means is that when the enzymes are done doing their job, the enzymes have to spend a little bit of time looking for a new substrate to catalyze the next reaction. In scientific terms, when there's not enough enzymes or not enough substrate to keep the enzymes busy, this is called substrate exhaustion. Let's check out another environmental condition that affects enzymes. This is the effect of substrate concentration on reaction rate. You'll notice right away that the curve looks similar to the last one we saw with enzyme concentration. In fact, I just copied and pasted that and changed this word right here from enzyme to substrate. But let's just go with it. So to describe the effect of substrate concentration on enzyme function, just read the graph. On the left side of the curve, when you increase the substrate concentration, you also increase the reaction rate. But then at high levels of substrate over here, what ends up happening is the reaction rate plateaus, flattens out. So then at that point, adding more substrate won't make the reaction rate go faster. That's the describe part. Now let's explain what's going on at the molecular level to make that happen. To do the explain part, I really like the visuals of this diagram to help us out. Remember, for enzymes to catalyze a reaction, the enzyme needs to be near a substrate and collide with the substrate. So if there aren't that many substrates, like over here, what happens is that the chances for a successful collision are pretty low. There are enzymes that can help with a reaction, but these enzymes are simply empty, like this one, because they can't find a substrate. Look at this guy over here. He's pretty far from any substrate that he can collide with. But then, when more substrates are added, this increases the chances for a successful collision between enzymes and substrates, and as a result, more reactions are happening, increasing that reaction rate. At some point, though, adding more substrate won't cause any further increase in reaction rate. This is because all of the enzymes right here are full and they're working as fast as they can. As soon as the enzyme spits out this product and becomes empty, there's so many substrates that can fill that active site and do the next reaction. A substrate is within immediate reach for the next reaction. The scientific name for this condition is enzyme saturation. The enzymes are saturated full with substrates and can't work any faster. Our next factor is temperature. You'll notice that the shape of this curve is different than the previous two. So let's describe what's happening. On the left side of the curve, over here from the peak, an increase in temperature causes an increase in reaction rate. But after the reaction rate peaks, the right side of the curve shows us that the reaction rate will quickly decrease with increases in temperature. The point where the reaction rate is fastest is called the optimal temperature. Now that we've described the what, let's explain the why. The left side of the curve can be explained with kinetic energy, the energy of motion. When molecules are heated up, the molecules move around faster. They have more kinetic energy. When molecules are moving around faster, the chances for collision increase, causing this increase in reaction rate. Somewhere in the middle, we hit the optimal temperature. This is when there are the most successful collisions between enzyme and substrate. But you can see that once the temperature increases over the optimum temperature, bad things start to happen. And we can explain this by talking about how molecules move. Besides molecules moving around and colliding with one another, molecules actually have internal movements inside of their structure. So when more heat is added, the internal molecules movement starts to increase to the point where the molecule actually unfolds and changes shape. Now, we've heard of this before in our biochemistry unit, and we called that denaturation, when proteins unfold and change their shape. Now, with denaturation, proteins become so unfolded that it prevents the protein from doing its job. Now, if you remember what we talked about with enzymes, enzymes depend so much on their shape to properly function in their active site. So changing an enzyme shape by denaturation prevents the substrate from even binding in the first place. And this denaturation is what causes this reaction rate to go down so much. I really like this graph because it shows us a great connection to evolution. Enzymes in different organisms that live in different conditions can work at different temperatures. This one on the left shows a human enzyme. Humans have a body temperature of around 37 degrees centigrade, so most of the enzymes in our body have an optimal temperature right around there. But there's other organisms that live at other temperatures. For example, thermophilic or hot spring bacteria, they couldn't have enzymes with an optimal temperature that's so low. You can see over here that the reaction rate would be 0. Instead, these bacteria have evolved to have enzymes that can withstand and function at these high temperatures of about 70 degrees Celsius. Next is the effect of pH on reaction rate. Like temperature, there's a sweet spot right here, an optimal pH, with a maximum reaction rate, but pH that's higher or lower than the optimal rate will have a decreased, or optimal pH will have a decreased reaction rate. Now let's explain why this is. The reason why having a pH that's too high or too low over the optimum is bad connects directly to our biochemistry rule of how like charges repel and opposites attract. So how pH works is pH adds or removes hydrogen ions from amino acids. You can see in this diagram the same amino acid at a low pH, neutral pH, and high pH. And what ends up being different is at a low pH, there's this hydrogen. At a neutral pH, this hydrogen is removed. And at a high pH, this hydrogen is removed. And you can see that there's this big charge difference. You get a negative charge here, but then you lose a positive charge. So these changes in charges of amino acids can affect the local folding and global folding of peptides, of proteins. And then this change in folding is denaturation that can lead to a change in structure that decreases the reaction rate because substrates will no longer fit in the active site of the enzyme. What's cool about enzymes and pHs is that different enzymes have a different optimum pH. And there's a really neat example in our digestive system. In our digestive system, there's different regions that have different pHs. Our stomach makes stomach acid. That makes the pH really low. So the enzyme that works in the stomach, called pepsin, has an optimal pH that's really low. By the time the food that's in our stomach exits to the small intestine, the pH gets neutralized. The enzyme that's most active in the small intestine is called trypsin. And you can see that trypsin has an optimal pH that's higher, about 8.5. So now I want to talk to you a little bit about cofactors and coenzymes. We're just going to touch on this. So what cofactors and coenzymes have in common is that both of them increase the efficiency of enzymes. And what's different about cofactors and coenzymes is just kind of like how they look and what they're made up of. So cofactors, are small and inorganic, whereas coenzymes are a little bit bigger and organic. Some examples of cofactors are all of these ions that are here: magnesium, calcium, zinc, iron, copper, and potassium. And how they work is that they're bound within the enzyme and they help the enzyme work. So you can see an example here is with magnesium inside of chlorophyll. So magnesium is that little guy right in there. Or how iron helps in hemoglobin. You can see 4 iron molecules inside of hemoglobin. Coenzymes that are a little bit bigger bind the enzyme near the active site. We'll see some examples of coenzymes that include a molecule called FAD and another one called NAD. They're going to be the main ones that we're looking at. For the last part of these notes, I want to look at inhibitors. Inhibitors are small molecules that decrease the efficiency of enzymes. They inhibit. So cells can actually more easily use inhibitors rather than changing their pH or changing their temperature to try to shut an enzyme off or take an inhibitor away to turn an enzyme on. So think of inhibitors as kind of like molecular switches that can regulate enzyme function. A lot of drugs actually also can act as inhibitors for enzyme. Now, we're going to classify inhibitors as either competitive or non-competitive based on their mechanisms of action. So let's jump right in to the difference of how those two work, competitive and non-competitive. A competitive inhibitor gets its name because a competitive inhibitor competes with the substrate for binding to the active site of an enzyme. In this diagram, you can see that the enzyme is purple, the inhibitor is blue, and the substrate is this pink color. the substrate can't get into the active site of the enzyme because there's an inhibitor there. An inhibitor is blocking the substrate from ever getting in. What we can assume from this, knowing what we know about the active site of the enzyme, is that the inhibitor that's in here and the substrate have to have pretty similar shape to match up with the active site of the enzyme, and have to have pretty similar chemical characteristics to match up too. Another way that inhibitors work is by non-competitive inhibition. In non-competitive inhibition, the inhibitor and substrate don't compete for binding to the active site. In this case, you can see the enzyme is purple, the inhibitor is this yellow color, and the substrate is red. How this works is that there's a binding site on a different side of the enzyme. And this different site of the enzyme is called an allosteric site. Now, when there's an inhibitor sitting in the allosteric site of the enzyme, that binding between the inhibitor and the enzyme changes the shape of the active site. And now, the active site isn't in a favorable shape to accept a substrate. So now, the changed shape of the active site of the enzyme inhibits a substrate from binding. In other words, binding of the inhibitor causes a conformational or shape change of the active site. One thing that you'll see once you start to understand this and get kind of like these templates down is you'll see that biology takes these simple templates and does things with them. So allostery, where a regulator molecule, an inhibitor, binds to a different site besides the active site, that allostery can actually be activating or inhibiting. So what you can see in this case is that an inhibitor binds in a site outside of the active site, just like we saw in the last slide, and prevents a substrate from ever even getting in. But another thing that can happen is that an activator molecule can bind to the enzyme and cause the active site to stabilize and stay open. And now in this case, we have allosteric regulation, but it's allosteric activation. So I want you to make sure to keep an open mind here. And when you see something new that seems a little bit different than the templates that we're studying, just adjust your framework a little bit and be open to the beauty of biology. So let's recap here what we learned about things that can affect an enzyme function. The environmental effects of enzymes are substrate concentration, enzyme concentration, temperature, and pH affect the speed of enzymes. It's easier for cells to regulate inhibitors, so a lot of cells use inhibitors to turn an enzyme on and an enzyme off. What you should be able to do with substrate concentration, enzyme concentration, temperature, and pH changes, you should be able to describe the effects of each of these on reaction rate of enzymes. But in addition to describing, you should also be able to explain why each of these things affect our reaction rate. Next, a significant amount of time was spent explaining how inhibitors can block substrates by entering the active site, by competitive inhibition, or by changing the shape of the active site with non-competitive inhibition and allostery. If you have any questions about this, be sure to ask in class. We're going to get some more repetitions of this, so you don't have to be a master yet, but things should start falling into place by now.